Okay, I think we're going to get started on our AACR Outstanding Investigator Award for Breast Cancer Research talk today. So I know it was a very exciting session before this, and it's been a packed day so far. So good afternoon. I'm Hope Rugo from the University of California, San Francisco. I hope everybody's been enjoying the meeting. And today I have the great privilege to introduce the 16th recipient of the AACR Outstanding Investigator Award for Breast Cancer Research. Since 2008, AACR has been distributing this special award to recognize investigators of no more than 50 years of age, we give up people's ages with this award, whose novel and significant work has had or may have a far-reaching impact on the etiology, detection, diagnosis, treatment, or prevention of breast cancer. Such work has involved numerous disciplines across the continuum of biomedical research, including basic translational, clinical, and epidemiologic studies. I'd also like to take a moment to thank the Breast Cancer Research Foundation. As you may know, BCRF's mission is to prevent and cure breast cancer by advancing the world's most promising research. Since 2018, BCRF has generously supported this special award, which continues to provide an important platform by which to recognize and honor emerging leaders in the breast cancer research field. Please join me in thanking the Breast Cancer Research Foundation for their ongoing support. This year's award recipient, Dr. Alana Welm, continues the rich tradition of this award in recognizing impactful scientific achievements in breast cancer research. Dr. Welm is currently Senior Director of Basic Science at Huntsman Cancer Institute, Professor in the Department of Oncological Sciences, and the Ralph E. and Willia T. Main Presidential Endowed Chair in Cancer Research at the University of Utah. Dr. Welm is being recognized for her innovative and impactful research dedicated to the understanding of fundamental biology of metastatic breast cancer and for translating these findings into new and life-changing therapeutic approaches. As many of you know, the vast molecular heterogeneity of metastatic breast cancer has often limited the effectiveness of state-of-the-art genomic technologies in predicting biology and/or informing treatment. Dr. Welm has aided in remedying this issue by developing innovative human breast cancer models that accurately reflect metastatic behavior and treatment response. Her group recently demonstrated the power of such patient-derived models to recapitulate metastatic breast cancer and optimize patient-specific therapy in real time. These models are now widely considered the gold standard for breast cancer models and have led to funded clinical trials as well. Dr. Welm's outstanding work has been recognized in many settings, including by the University of Utah this year when she received one of the university's Distinguished Research Awards. She's also an internationally recognized leader within the breast field and serves on a number of different committees with the NCI and DoD and is the editor for breast cancer research as well. So you don't want to hear from me, you want to hear from Dr. Welm. So I am going to, let's see here, move on and tell you just at the moment that she's also on the board of directors of the Metastasis Research Society as president-elect and has been on our program committee, the AACR Annual Program Committee, and the AACR Tumor Microenvironment Working Group Steering Committee. So Dr. Welm, who is an exceptionally talented and internationally recognized scientist who has applied her research discoveries to the development of improved breast cancer therapies, is widely known for her creativity and impact, is a valued contributor to the community through her leadership, generosity, and collaboration, and I'm thrilled to announce her as this year's recipient of the AACR Outstanding Investigator Award for Breast Cancer Research. And Dr. Welm's award is sitting in that box, but we'll go ahead and listen to Dr. Welm now instead.
Thank you very much, Dr. Rigo, for that fabulous, very generous introduction. Thank you to the AACR and the BCRF for this great honor, which really I'm happy to accept on behalf of my whole team, which is quite extensive. A lot of people contributed to this work. So our team is very multidisciplinary with Brian Welm, my partner at home and in the lab, as well as a really important clinical team that helps us translate our research to the clinic, even when we have some unconventional ideas. One of the things that I realized as I was putting this talk together is we actually have, we do more modeling in our family maybe than most, including some metal sculptures of scientific models that Brian does in his spare time, and I thought people might like to see this estrogen receptor dimer with tamoxifen bound to it. So here are my disclosures. None of them are relevant for this talk. I'm going to be talking about the main motivation of our lab, which is really the fact that metastatic breast cancer is still incurable. So despite state-of-the-art therapies, earlier diagnosis, advancing treatments all the time, once the disease recurs, which is in about 20 to 30% of cases in the distant site, the disease is still more and more treatable, but incurable, and that leads to more than 40,000 deaths every year just in the US. So there's a lot of therapies approved for advanced breast cancer, which are growing all the time. We hear about many of them here at this meeting. One of the things I want to point out, though, is that all these drugs, remember, are approved because they do have efficacy in the population. But I think from an individual patient perspective, it's really unclear which drug might work best for their tumor because of the heterogeneity of metastatic breast cancer. So how do you choose from this long list of therapies? All of these therapies have toxicities, some more than others. And so one of the things that motivates us is trying to understand whether we can do a better job in matching the approved therapies to the tumors that will benefit most from them. And this, of course, is the whole point of precision oncology, which has been a very exciting advance in many, many types of cancers. And we do have a number of drugs that are approved based on the presence of mutation in breast tumors, shown here in red in the list above. But what we want to move to is actually using functional drug responses in more advanced patient-derived models to really hone in on which therapies are going to be best for which tumor. And for that, we need to build better models. The idea is that in the long term, we hope this would have potential impact for individual patients where their tumor might receive a drug that will be more likely to be effective and also not receive the therapies that are not effective and hopefully spare certain toxicities. So we really believe that we need combination strategies in order to improve outcomes for breast cancer. And this would depend on both targeting the metastatic breast tumor themselves, but also the supportive tumor microenvironment. So I'll be talking a little bit about what we're doing to model the metastatic breast tumors using patient-derived xenografts or PDX, as well as organoids, where we hope to use them to help develop new drugs, identify effective combinations, and use them in a way that is precise, using the right drugs for the right tumors. And then on the flip side, targeting the tumor microenvironment, I think we're all still very excited about immunotherapy. I think there's still a lot of promise in this area that is unrealized. And here we use genetically engineered mouse models to best approximate the tumor microenvironment. So first, let's focus on the tumor side. So for the last almost 2 decades, my lab has been generating patient-derived xenografts from breast cancer patient tumors, and now also organoids. And I'll note here, this is just a list of our published models, although we have many more coming out all the time. We're able to make PDXs of all clinical subtypes of breast cancer now, although I'll say that TNBC is overrepresented because they're easier to build. But in my lab, we try really hard to make models from metastatic disease. And you can see on the bottom row there that we've collected metastases from all different tissues, both from living patients undergoing biopsies or surgeries, but also in our new Legacy to Life rapid autopsy program. And then we try to develop models from patients over time. So highlighted on the top are some doublets or triplets where we've made models from the same patient over the course of her therapy in order to try to use those to model tumor evolution and acquisition of drug resistance. Now, PDX models have many advantages that I won't go into. I think in general, the field thinks they're a pretty good representation of human tumors. They're not perfect. No model is perfect. Some are useful. I can't remember who said that, but it's very true. What I think is an advantage of these models is that the ER-positive tumors do maintain their estrogen dependence in PDX as well as their endocrine resistance. So you can use those to study some of those very important problems in the majority of breast cancers or metastatic breast cancers that are ER positive. They also exhibit spontaneous metastases in these mice, which is useful. That's actually the reason I made them in the first place. There are disadvantages, though, too. So the most obvious one is that the hosts are immunocompromised in order to allow the tumors to grow. So this prevents us from studying some of those important tumor-immune interactions. But they're also quite slow and labor-intensive. Especially ER-positive breast cancers can take more than a year to develop. They grow slowly, just like they do in humans much of the time. So they're not really appropriate for high throughput or that kind of study. It's better for sort of low throughput validation studies. And that's the reason why we decided to develop organoids. And our strategy in our lab was to develop an organoid bank from our PDX models. And the reason for this was we liked the idea of having a matched in vitro and in vivo pairs of the same disease. So we could do some of the more feasible high throughput stuff in organoids, but then go back into the matching in vivo model and ask whether the drug or whatever strategy we had developed would actually shrink tumors, because that's what we need to do. So the advantages of this, I think, are the feasibility of organoids, although it did take us quite some time to develop and optimize this. And I want to say that, I'm highlighting people in the lab who've done this over the years, and I'm really, really proud of my team for developing this and sticking with it. One of the features of our PDX-derived organoids, or PDXOs, is that we've removed the mouse stroma. And we've done this because our goal was to generate a large bank of human breast cancers that were relatively uniform. And we had some trouble with some of the stroma sometimes taking over the culture. So while I recognize that's a caveat, because we know tumor stroma interactions are important, for the purposes of this study, we did remove it. So now we have a collection of breast PDX with matching PDXO, full genomics, and de-identified clinical data. And we have already published their fidelity to the human tumors that they came from. And if you're interested, please feel free to look at that paper. So the next step then is not advancing. Let's see. There we go. What are we going to do with this big collection of models? So together with Baylor College of Medicine, Mike Lewis in particular, we are part of the NCI's PDX Net, which is a PDX development and trial center. And the goal of our grant was to develop these models and then use them to screen drugs. And these drugs would be drugs that are already in the NCI CTEP program, so they're ready to go in humans if we could find a good application. So we realized that we could use our organoids to do a relatively medium throughput screen in 384 well format. And because this hadn't been done before, we really wanted to make sure that it was reproducible, so we did the whole screen in 3 biological replicates using the organoids at different passages after they were developed. And then, of course, we validate anything interesting back in the PDX model. So we've published the pilot screen here. The rest of the data is still unpublished because we're working on it. But the big picture goal here was we made 100 breast cancer lines. So in combination with Mike's PDX models, we could, in my lab, generate 100 lines through really hard work from my team during COVID even. And then screen all of them with about 50 drugs from the NCI CTEP program. And then the idea is that now you would see which drugs are working in which types of breast cancer, irrespective of clinical subtype, and together with the omics, try to understand why they're working and find a biomarker to develop better clinical trials. So just very quickly, I want to set the landscape for how we did this screen. So our screens are done on mature organoids, not on single cells. So these organoids are plated in 384 well format in 2 plates. One of them is the screening plate that will receive drugs at different doses and in a quadruplicate replicate. And the other is what we call a day 0 plate. This is a plate that allows us to assess the growth rate over the course of the assay. And that's because in our lab we realize every single breast tumor has its own personality, and their growth rates depend, their drug responses do depend on their growth rates. So as they're screened over a 6-day assay, the drug screening plate measures efficacy of the drugs. We also measure growth rate, and then we can put that together to basically come up with a growth-adjusted efficacy, which is an area over the dose-response curve. Now, there are some limitations to this assay that I'll just highlight here. One is that we're screening in 6 days, so anything we can't see in 6 days is not going to come out. And also, we're using cell titer glow 3D. That means we're measuring ATP content as a surrogate of cell viability in these assays. So the idea is to take our 100 PDX, make 100 PDXO, stable cryopreservable lines, screen them with drugs, identify the responders, and then adjust that with the growth rate so that we can determine the growth-adjusted drug responses. And then together with our collaborators at University of Utah, Dr. Gabor Marth's lab, who are computational scientists, integrate all of that data with the omics of the model in order to, and machine learning approaches in order to come up with the patterns. This was a big project, and so we broke it into 4 parts. We did screens A through D, where each screen comprised 25 models. The composition of the models in each screen is shown at the bottom. And then we started with 50 drugs. At the end of 25 models, we assessed whether the drugs looked promising or not. Some of them we dropped out, either because of undue toxicity or because they didn't have efficacy. And then, of course, if new drugs came along, we added those in. So by the end of this project, we had screened 100 models with a total of 94 drugs, again, in 3 biological replicates. And this was the composition of the breast cancers that were screened. So this is not meant to be read. This is just a picture of the type of data we're looking at. So as you can see, this really has turned into a data science problem. And thank goodness for my computational science collaborators. We've generated nearly 500,000 data points of drug response in organoids and 1,700 or so data points for validation experiments in PDXs. Here I'm just showing you, we don't have screen D here because it's relatively recently completed. But what you can see is that with unsupervised clustering, we can identify patterns of models, clusters of models that respond to different types of drugs. If we look at the drugs that were non-dropouts, so they were carried through the entire models, now this increases our power to find drugs that look promising across a large number of models, and we can start to parse out who are the responders and non-responders, and also look at overlapping sensitivities, which hopefully can give us a hint of combination therapies to try next. And that's our goal for the next 5 years in our PDXNet project. So what did we find? We're actually in the middle of still analyzing this data, so I don't have any really exciting things for you except a few hints. So here is one of the findings that we had early on that was really quite interesting to us. This is a SMAC mimetic called birinapant, so it's an inducer of apoptosis. And we found that we were seeing some nice cytotoxic activity across a panel of organoids, but with selectivity. So some models are responding really well, as shown with this tall red cytotoxicity, even at very low doses of the drug. And this was happening across different subtypes of breast cancer. When we went to our in vivo matched models, we could see that some triple-negative breast cancers responded really nicely with complete responses to this single agent, which was quite remarkable. Whereas other triple-negative breast cancers are growing just happily in the presence of this drug. So this really indicates we need a biomarker for this. We also know that any drug is probably not going to be sufficient in metastatic breast cancer, and so we've been combining this with chemotherapy, where we can see that after a short period of treatment, when we get responses, if we follow these mice over time off treatment, we can measure the durability of response. And here's just one example where this drug's been combined with chemotherapy, and off treatment, we see a statistically significant improvement in overall survival of the mice, indicating durable responses. So now we've written a clinical trial to test SMAC mimetic in combination with chemotherapy in metastatic triple-negative breast cancer. We're very excited about getting that started. And then Elizabeth Brown, a grad student in the lab, is working really hard to determine the mechanisms of response and resistance to this drug, which will hopefully refine biomarkers for future trials. In the meantime, what else have we learned? One of the observations we made serendipitously was that engraftment of the PDX, which is not easy in breast cancer, correlated really well with outcome of the patients. And this was an observation we actually published more than a decade ago when we made our first PDX paper, where people whose tumors could grow in our mice had poor outcomes, whereas people whose tumors did not grow had good survival times from breast cancer. And this was independent of any other clinical variables, which really piqued our interest. This was retrospective data, so we decided to open a prospective study. Here we enrolled newly diagnosed, non-metastatic, ERPR negative or low patients, so they had to be less than 10% ERPR positive. And we biopsied their breast tumor before they started therapy. We asked whether they could grow in mice, and we asked whether that would correlate with patient outcome. So this study is now completed, and we're continuing to follow these patients. We enrolled 80 patients. You can see that 61 of them were TNBC or TNBC-like, meaning they were less than 10% ERPR positive, but still HER2 negative. We had 18 HER2 positives and 1 mixed. The engraftment rates of these tumors were just as we expected from our previous experience. The patients, while we were busy engrafting their tumors and waiting for them to grow in mice, they underwent their standard uniform neoadjuvant chemotherapy that was matched to the subtype of cancer they had. And then at the end, when they underwent surgery, we were able to assess whether or not they had complete pathological response or whether they still had residual disease. So what did we find in terms of PDX engraftment and correlation with patient recurrence? First, I'll show you the correlation with PathCR. So we see a trend in our study of patients who have residual disease in their tumor after chemotherapy have a trend toward more recurrence. This is something that's been demonstrated for a long time with TNBC, and our study is just not powered with 80 patients to see a significant effect here. And of course, we're following these patients over more time. But what the study was powered for, and what we did in fact find, is that the PDX engraftment does correlate very strongly with both recurrence-free survival and poor overall survival. So patients whose tumors engrafted had more recurrences, and when they recurred, they had uniformly fatal disease. So all of these patients died within about a year after their surgery, this really suggests that these PDX engraftments are really picking out the most aggressive disease. So our median follow-up time at this point is 2.3 years from surgery, and you can see that even already we see very strong hazard ratios for PDX engraftment predicting reduced breast cancer-specific survival. And one thing that I don't have time to show you, but when we do subset analysis, the effect really is in the triple negatives. It's in the HER2 negative population. So these patients have very aggressive tumors that we can identify ahead of time by PDX engraftment, and yet they're receiving state-of-the-art therapy and dying of their disease. So this is a population that we really need to understand better, and the question is, what can we do for these patients? Well, we hope that our discovery science that I just talked about will lead to new drugs that are effective in these, and I will note that one of the drugs that we're excited about was very effective in 4 of these patients' tumors, shown with the blue arrows. So we have hope that we can find drugs that work even in these very aggressive tumors, and that will be the point of this trial. But what else are we doing? So we've started a follow-up to the TOWARDS trial called TOWARDS 2, where again we enroll newly diagnosed triple-negative breast cancer patients, or TNBC-like with low ER. We again biopsy their tumor before they start their neoadjuvant therapy. Now we not only make the PDX to predict recurrence, but we make organoids and screen drugs. And the idea here is that if and when they have recurrence, we can actually return the results of that drug screening through a functional precision oncology tumor board to hopefully personalize their therapy earlier on before the disease gets too advanced. We have a very similar trial open also in metastatic breast cancer, where we don't need PDX to predict recurrence. That's already happened. So here we go straight to organoids and drug screening and do the same thing, returning results after they've completed their first line of standard care therapy in the metastatic setting. This is a hard problem. These are challenging trials to do, and it's something that the Society for Functional Precision Medicine is thinking a lot about, and I encourage you to follow them if it's something that interests you. So we've already published some of the promising results with this concept, but I still think there are major limitations. One of them in our trials is that we're not a CLIA-certified lab, and so the IRB restricts us from returning any results for drugs that are not already FDA approved for breast cancer. So essentially, these trials are feasibility studies and are able to hopefully prioritize approved drugs for patients, but we really want to get to the point where if this works, we can use this to screen much wider swath of drugs because, as I showed you already, the efficacy of our available drugs in breast cancer right now is limited. All right, so we've talked about the tumor side. Let's switch over to the tumor microenvironment, where we're very excited about immunotherapy's promise. You know, we have this huge bank of PDX models, and our first thought was, well, let's humanize the immune system and create humanized models. And we have published a paper that this, of course, is possible. Other people have shown this as well. But in our analysis, and you can go into that more in this paper if you're interested, we really don't think that it's really physiologically relevant enough to recapitulate the tumor-immune interactions in the way that we want. So for this, we've turned, again, to genetically engineered mouse models to answer this question. So I'm going to tell you now about a really, I think, an exciting and new immunotherapy target that we've been working on called the RON receptor tyrosine kinase. So RON is a single membrane receptor tyrosine kinase that gets activated through its ligand, a macrophage stimulating protein, that induces receptor dimerization and signaling. We've been working on this for quite some time and have shown that in tumor cells, RON kinase promotes metastasis, and it's actually sufficient to convert non-metastatic breast cancers to metastatic. And that's through its signaling through PI3 kinase, AKT, and mTOR. We also have found that in immune cells, RON kinase regulates the anti-tumor immune response, and because this is a kinase, it is targetable, so it does have therapeutic potential. I'm going to show you just a little bit of that data here, just to put everyone on the same page. So here we're using genetically engineered mouse models, the MMTV-PYMT model, first developed by Bill Muller, as well as a P53 BRCA1 knockout model that was given to us by Chuck Peru. And in either spontaneous metastasis or experimental metastasis models, which is what's shown here, we see that mice that lack the RON tyrosine kinase domain in the host are protected from metastasis. And we can do this genetically, but we can also do this in wild-type mice by treating with a RON kinase selective inhibitor after the metastases are seeded and growing, and again, get protection from metastatic outgrowth. Our first hint that this was involving the immune system came when we depleted CD8 T cells in the same experiment and found that now we lose the efficacy of the inhibitor, really suggesting that the protection from metastasis was involving the immune system. So that led us to think about RON inhibitors as new immunotherapy targets. And there's a number of ways to inhibit this kinase. One would be to use anti-RON monoclonal antibodies that could either block receptor dimerization or ligand binding. And the other, as I just showed you, would be small molecule tyrosine kinase inhibitors that can be selective for this kinase and serve as ATP competitive inhibitors. So we needed to decide what would be the right therapeutic strategy, and for that we went back to the science. And this was important because the RON gene actually encodes a number of different RON isoforms. I've already told you about the full-length isoform, which is encoded, shown here. But there's also a short form of the RON kinase that gets initiated through an alternative promoter in intron 10 of the gene. And this generates a protein that is completely in frame, but is lacking the N-terminus. So it's lacking the ligand binding domain, it has just the transmembrane and kinase domains, and it has constitutively active kinase activity. So the question for us, it was which isoform is important? Because if the short form is important, then an antibody-mediated strategy is not gonna work. So we felt we needed to go back to the lab to figure that out. For this, we utilized a number of different mouse alleles that were made by Susan Waltz's lab and have been studied mostly in some cancer settings and also inflammatory immunology settings. So we have wild-type mice that, of course, can make both isoforms. We also have the tyrosine kinase knockout that I showed you already, in which the kinase domain is deleted, rendering both isoforms inactive. And then we have a short-form RON-only knockout mouse. And the way this was made is by knocking in the cDNA of the second half of the gene into the endogenous locus, which eliminates this promoter that's in the intron. So these mice can make full-length RON. It's perfectly functional, but they can't make any short-form RON. So next, we asked in these mice what happens with metastasis. To our great shock, we found that just deletion of short-form RON only was sufficient to protect from either lung metastasis, shown here, also bone metastasis, shown here. And so this really said, wow, it's the short form, the one we thought we'd rule out, that is the important isoform. Now, why are these mice so protected from metastasis? It turns out it's because they can metastasize spontaneously. Once they're in the lung or the bone, they can start to grow, but about 2 weeks later, they get swarmed by T cells. And this is both CD4 and CD8 T cells, suggesting that they're being protected by metastasis because they have a stronger immune response to these metastatic disease. So to get into this mechanism, we decided to do single-cell RNA sequencing in the lung metastasis model during this metastatic outgrowth phase. So we isolated the immune cells using CD45 and then did single-cell RNA-seq. We thought we'd see T cells, CD8 T cells would be the culprit here, and in fact we did see a difference in T cells, but to our surprise it was a CD4 population that was different in the short-form RON knockout mice. And even more interesting, the CD4 T cells were unique in that they're still expressing TCF and LEF as well as CCR7, which indicates that these are naive or stem-like CD4 T cells. A corollary to this has been described to be protective in tumors in the CD8 population in a different kind of cancer, so we thought this might be important. When we look at these micrometastases that are getting cleared, so again, in the short-form RON knockout, these get swarmed, and in fact, it is the TCF-expressing CD4 positive T cells that are swarming these tumors. So to ask how important they were, we depleted CD4 T cells and did the same experiment. And what you can see is that loss of CD4 T cells in this model now allows for metastatic outgrowth, and so there's significant rescue here. This got us thinking about the CD4 helper function, where the main role of CD4s can be to help CD8 cytotoxic T cell responses, so we looked at that. I don't have time to go into all the tools and tricks we had to build for this, not being in a C57 Black 6 genetic background, we're an FVB here. And so we had to build a lot of tools, but essentially we finally got tumor-specific tetramers and peptides that we could use to assess the immune response. And indeed, we can see that the short-form RON knockout mice, when challenged with tumors, have more anti-tumor specific CD8 T cells, making interferon gamma. We can detect that with tetramers as well. And importantly, they have more effector memory cells. And this is important because in tumor re-challenge experiments, we have seen that the short-form RON knockout mice have better memory and can eradicate tumors months after they have eliminated the first set. If we deplete CD8s, again, we rescue metastasis. So both CD4s and CD8s are important here. So just to summarize what I've told you, the short-form RON knockout mice are protected from metastasis in both lung and bones. And in the lung, what we see is that the tumors get infiltrated by these TCF-expressing naive or stem-like CD4 cells, which I didn't have time to show you, but are sort of skewed to differentiate to a Th1 type phenotype, which can enhance CD8 T cell cytotoxic activity. This is in contrast to the normal setting where the tumor microenvironment is such that these cells are really skewed toward a Th17 and Treg phenotype, which is immunosuppressive in this setting. So what's the clinical relevance? We're hoping now that we can use RON kinase inhibitors because we have to hit the short form. And this would do, we're still unsure what RON is doing to increase these TCF1 expressing CD4s. That's an active area of interest in our lab. And why it is that these are so skewed toward a Th1 phenotype. We hope that these RON inhibitors could potentially prevent metastatic outgrowth like they do in our mice if given in, say, an adjuvant setting. We hope that they might have long-term protection from future relapse because of the nice memory phenotype that we've seen. But we also have to ask, what about already established metastases? Could these inhibitors actually boost the immune response and eradicate existing metastases? RON inhibitors have been in the clinic. There have been several of them already tested in phase 1 and phase 2 studies. This is a study we did in collaboration with Aslan Pharmaceuticals, where we were able to show that the drug was safe and it was well tolerated. And here's a pharmacodynamic marker looking at bone turnover. And the reason we looked at this is because RON is involved in the bone osteoclast. So we were able to show that at a safe and tolerable dose, we could in fact inhibit RON in patients. So we think this has some promise going forward. Can we eradicate existing metastasis? That's raising the bar a little bit, but we decided to try it. So again, we have mice that have already established metastases, and we treated them with either the RON inhibitor alone, immunotherapy alone, which we used either anti-CTLA-4 or anti-PD-1, or of course the combination. And here we try to control the specificity of the inhibitor. These ATP competitive inhibitors are selective for RON and also MET, another member of the family, but they do hit other kinases. And so we controlled for that by using the tyrosine kinase knockout as a host control. What we found was that actually the combination of the RON inhibitor and anti-CTLA-4 gave a very high clinical benefit to these mice, meaning about half of the mice had partial responses and the other half had complete responses. Interestingly, the combination with anti-PD-1 did nothing. And so we're still investigating why is that? What's the biology behind the selective combination in different immunotherapies? So to end here, I just want to recap that blocking short-form RON specifically prevents from metastases through expansion of the CD4 population. Short-form RON is expressed in these CD4 T cells, and it seems to be regulating their proliferation and differentiation. We're still working on that. We have to target this using kinase inhibitors, not extracellular antibodies, and these may work in combination with existing or maybe other new immunotherapies. I think just finishing everything, I would say that our strategy of using combinations of patient-derived models to try to find good targets in human tumor cells acquired from patients in the metastatic setting, post-treatment, and a combination of using genetically engineered mice with metastatic tumors, where we can best approximate that tumor immune microenvironment, we think is a good strategy to try to advance the field. So with that, I want to stop and thank everybody who did the work. Here's a relatively current picture of the lab, although many, many past members have contributed to all this foundational work. I want to thank our patient advocate team at Huntsman, who are just fabulous to work with and great inspiration. Our Legacy to Life program is a new rapid autopsy program. I want to thank all the participants and the families who participate, because that's one way we can get these metastatic post-treatment really awful tumors in order to study them better. We have so many great interactions. I already mentioned the PDX-NET, but the NBCC Artemis project has been a great source of inspiration for really doing that hard experiment, even though it sounds crazy. I mentioned some of the others here. I want to thank our funding. And then I just, in the end, want to just plead with everyone to please share your data, share your models. I mean, all of us are working hard in our labs. We're all making important scientific discoveries that we hope advance the field, but any one of us is not going to be enough to really make a difference for people, which is what we're trying to do. So we all have to put our heads together and do it together, ask the hard questions, and get it done. Thank you.
Turn it on. There we go. Thank you, and congratulations on this well-deserved award. I really appreciate it. We don't have time for questions, and the room is now open for the next session, so thanks again.
Thank you very much.
Congratulations.
Okay.